Hello everyone, myself Shani. I am currently pursuing my MSc degree from Tripura University. Today we gonna learn about restriction endonuclease basics. So before we start restriction enzyme, we should know about the nucleases for better understanding. What is nucleases? Nucleases are group of enzyme that act on genetic material by cleaving the phosphodiester bond of nucleic acid. It can cut DNA, it can cut also RNA. Basically, we can classify nucleases into two categories. One is exonuclease, another one is endonuclease. Exonucleases are those nucleases that remove nucleotide from the end of polynucleotide chain. It can be 5' to 3' direction or 3' to 5' direction. In this figure, we can understand that nucleotide has been removed one by one by exonucleases. So now, what is endonucleases? Endonucleases are enzymes that cleave the phosphodiester bond within polynucleotide chain. It can cut DNA or RNA either specific sequence or non-specific sequence. What does that mean? Endonucleases are those nucleases that can cut genetic material in the middle of the polynucleotide chain. It can be specific sequence or it can be a non-specific sequence. So far now, what we know that nucleases are those enzymes that act on genetic material and cut the genetic material. Basically, there are two types. One is exonuclease, another one is endonuclease. Exonuclease can remove nucleotide from the end of the DNA or RNA molecule, and endonuclease are those that can cut genetic material middle of the polynucleotide chain. It can be specific sequence or it can be non-specific sequence. We should know the basic differences between exonuclease and endonucleases. First one is exonuclease always remove nucleotide from the end of the polynucleotide chain, and endonuclease can cut DNA at the middle of the polynucleotide chain. The second one is exonuclease are N-specific enzyme. It means it will remove DNA from the end like 5' to 3' end or 3' to 5' end. Endonucleases are site specific or sequence specific. It means these enzyme are cuts DNA at some sites or sequences. Third one is exonuclease can form sticky ends and endonuclease can form both sticky and blunt ends. What is sticky end, what in blunt end, it will come later. Example of exonucleases are exonuclease 1, exonuclease 2, and example of endonuclease are EcoR1, BMH1. So we can start our topic restriction enzyme. Restriction enzyme, also called restriction endonuclease, a enzyme produced by certain specific bacteria that can cut DNA into fragments at or near specific sequences, and those sequences are known as restriction site or recognition site. What does that mean? It is a type of endonuclease enzyme that recognizes sequence on a DNA, then it can cut the DNA at that specific sequences or near that specific sequences. The first restriction endonuclease enzyme was discovered in 1970 and the enzyme was Hint2. Now, why this enzyme are called restriction enzyme? Why the name is given so? The name is given because when certain viruses are attack the bacteria, this enzyme are protect the bacteria from virus and restrict the growth of viruses by degrading viral DNA without affecting the bacterial DNA. So what it is doing? It is restrict the virus growth in bacteria. That's why we call them restriction endonuclease enzyme. Now one question may come your mind. We know that restriction endonuclease cut viral DNA. So it can also cut the bacterial DNA. But why it is not cutting the bacterial DNA? Or how bacteria protect their DNA from their own restriction enzyme? The answer is simple. Bacteria protect their DNA by a system called restriction modification system, by an activity of an enzyme known as methyltransferase. The specialty of this enzyme is it can methylate DNA sequences or DNA bases by adding methyl groups on these sequences or bases. We know how restriction enzyme works. First of all, they recognize the sequence, then they cut DNA into fragments at that sequence or near that sequence. In bacteria, methyltransferase methylates those recognition site. Now restriction endonuclease can't act on this sequence. So if restriction enzyme can't recognize the sequence, so how can it will cut the DNA? That's how bacteria protect their DNA from their own restriction enzyme. Our next concept is nomenclature of restriction endonuclease. Restriction enzyme named are based on the organism in which they were discovered. The first three letters comes from the genus and species. The first letter comes from genus and the second and third letter comes from the species. And the fourth letter comes from the bacterial strain. And the Roman number indicates the number of the order of restriction enzyme that discover in that particular strain. We can understand this with example, like in EQR1, E refers the genus Escherichia and CO refers the species coli. R refers the bacterial strain, that is RY13, and the Roman number 1 denotes that it was the first restriction enzyme that was discovered in that strain. Likewise, in HIN3, H refers the genus name Haemophilus and IN refers the species name Influenza, and D was the bacterial strain, and the Roman number 3 denotes that it was the third enzyme that discovered in that particular strain. On the basis of enzyme subunit composition, cofactor requirements, nature of their target sequence and position of restriction site, we can classify restriction enzyme into 4 groups, that is type 1, type 2, type 3 and type 4. Type 1 restriction endonuclease were first to be identified. It cut DNA at far away from recognition site. It requires both ATP and edumate to do its proper function. It can do both the restriction digestion and restriction modification. Type 1 restriction enzyme possesses 3 subunits called HHDR, HHDM and HHDS. HHDR is required for restriction digestion, HHDM is required for modification, and HHDS helps in specificity of recognition. The example of type 1 restriction endonuclease are EcoK, EcoB. Type 2 restriction enzymes are most common and useful restriction endonuclease. It is an important enzyme in recombinant DNA technology. It cuts DNA at restriction site or near the restriction site. It only needs magnesium to its proper function. It is an ATP independent enzyme and it can only do restriction digestion. When the type 2 restriction endonuclease cuts DNA, it generates sticky and blunt end. The example of type 2 restriction enzyme are EcoR1, ALO1, etc. Type 3 restriction enzymes are the intermediate enzymes between type 1 and type 2 restriction endonuclease. It contains more than one subunit and requires ATP for its proper function. It recognizes 2 separate non-palindromic sequences and cleaves DNA at 20 to 30 base pairs away from their recognition site. Type 4 restriction enzymes are more complicated. It recognizes and cleaves only the modified DNA, that is methylated, hydroxymethylated and glucosyl hydroxymethylated. The cutting mechanism of restriction endonuclease are different for different restriction endonuclease. Some endonuclease can generate sticky ends and some can also generate blunt ends. Sticky ends are those ends that always have an overhang DNA strand. These ends are very important in recombinant DNA technology because the enzyme ligase can easily join these DNA because of this overhanged DNA strand. And blunt ends are generated when the endonuclease cut the DNA into straight. You can see this picture and clearly understand how these ends are produced by the restriction endonuclease enzyme. Our last concept is palindromic sequence. What is palindromic sequence? A DNA locus whose 5' to 3' sequence is identical on each DNA strand. The sequence is same when one strand is read right to left and the other strand is read left to right. See in this figure, if we read the upper strand 5' to 3' direction after ANT nucleotide, the third nucleotide to 8th nucleotide is matched with lower strand 3rd to 8th nucleotide at 5' to 3' direction. What I say, that upper strand GAATTC is matched with lower strand GAATTC in 5' to 3' direction in both strands, and these sequences are known as palindromic sequence. Recognition site of many restriction enzymes are palindrome in nature. However, it can be inverted sequence or many more. Look, there are lots of restriction enzyme and you can't remember the recognition sites of every restriction enzyme, but you should try to remember some important restriction enzyme. I have given here some restriction enzyme and their recognition site. Like EQR1, its recognition site is GAATTC, and the star mark is indicate that it will cut the DNA between G and A. The recognition site is GAATTC, but it will cut the DNA between G and A. Like in ALU1, the recognition site is AGCT, but it will cut DNA between G and C.